Okay. So here's just a little video for lectures 25 and 26 for the people who couldn't make it. So for lectures 25, we're going to talk about lectures 25 and 26, we're going to talk about 4 different types of signal transduction: our G protein couple receptors, our receptor tyrosine kinase, our gated ion channels, and our nuclear receptors, right? So my favorite one is probably our GPCR, easiest one to understand in my opinion. So we have this trimeric G protein right here, G alpha, G beta, and G gamma. G beta and G gamma are structural. G alpha is the one that's really important. So you're going to see as we have our hormone bind to our receptor, we're going to start activating a cascade, which will eventually lead our G alpha to be activated by the replacement of GDP with GTP, which will activate our adenylyl cyclase, which will then eventually form our cyclic AMP, which will continue down that signaling cascade, right? And if we want to deactivate our G alpha, we're just going to use a GTPase, which is going to hydrolyze our GTP to GDP, which will bring that alpha subunit back into our G protein trimeric structure, right? So the important thing is that as soon as we make our cyclic AMPR camps, we're actually going to be able to use it with protein kinase A. So we need 4 camps to release this inhibitory sequence from those catalytic subunits. And once we have those catalytic subunits up and running, then we're actually able to phosphorylate different areas of our body, either a protein or a different molecule, right? So the amino acids that can be phosphorylated again are serines, threonines, and tyrosines, because those are the ones that have the hydroxy group. Then we're just going to shift gears and talk about our desensitization pathway. So if our cells are activated for too much, or too frequently, we're actually going to be able to form a desensitization to that certain hormone. So in this case, it's pretty much as soon as the desensitization occurs, our G beta and G gamma are actually going to bring in our bark, which will phosphorylate the receptor, and then our bar is actually going to move that phosphorylated receptor to the inside of the cell through an endocytosis mechanism. Now for our receptor tyrosine kinase, this is probably the most complicated one. You have to know a lot of these different pathways, right? The important thing here is that once something binds to the receptor, we're actually going to cause an autophosphorylation event, which will eventually lead to the phosphorylated tyrosines to dimerize, and this dimerization of the receptor will activate this whole cascade, right? The important things to know here, I would really focus on our ROS, our RAF1, MEK, ERK, and then our ELK1 and SRF, because these are the ones that are going to come back in biochemistry. So it's always good to know them already. But you are still responsible to know a lot of these different components of the pathway. So again, if you don't have the time, focus on the RAF, MEK, and downward. But just be aware that he could ask any questions about this pathway. Finally, for our gated ion channels, these are some of the easiest ones. So we talk a lot about our neurons in this case. So depolarization event is when our sodium channels open and there's an influx of sodium into the cell. And then a repolarization is an inhibitory sequence, which is when our potassium channels actually go through an efflux, so they leave our cells. The most important thing here is that our calcium ion channels are really, really important in activating those neurotransmitter vesicles. And when they're activated, we actually diffuse it into the synaptic cleft, which can continue the depolarization event to the other neuron. And finally, the easiest one is the nuclear receptor. So these are hormones that are hydrophobic, and they're actually able to diffuse into the cell membrane and into the nuclear membrane where there's going to be a nuclear receptor. And that's where they can actually act as a transcription factor. Okay. And as a transcription factor, they usually bind to something called a hormone response element. And that hormone response element is going to make proteins associated with a specific hormone.